Hello students, welcome to lecture 1 of week 7. So in the previous lectures we have looked at the link read sequencing and how the link reads can be used to improve the sequence assembly quality, and also in combination with other kinds of reads such as nanopore or basically link reads generally are Illumina reads, right, or PacBio reads, right. So they can be used along with the other long reads to improve the assembly contiguity. And the link reads also have some advantages because they link two distant regions of the chromosome or the your DNA that you want to sequence, the genome, right. So a combination of link reads alone and its usage with other data will definitely help in improving your sequence assembly, right. So today we will see that just a quick overview of what link read sequencing was all about, that we discussed in the 6th week lectures, and also to look at method to assemble the link read data after filtering and cleaning, and then also look at other methods of scaffolding that are available, and then we will proceed to the usage of link reads and see that how link reads, nanopore reads, and a combination of the three, and along with the RNA sequencing data, how it improved the genome assembly in terms of the contiguity, N50, or the number of genes, Busco scores, etc. Okay, so basically link read sequencing is done by 10x Genomics. There is one more method, TEL-NOC, but generally like 10x Genomics have been the most preferred, although they are having some issues right now with their technology patent issue, but still link reads have done a lot for the improving the quality of the assembly. So basically it happens in a chromium instrument, right, and this is the flow cell of link read where 8 samples can be done together. So basically the method is to fragment the DNA, and the fragments can come from any region, right. There could be overlapping fragments also, and then each fragment will be barcoded separately, right, and then proceeded by sequencing of each fragment separately with their respective barcodes, and later on constructing an assembly. So it helps to also resolve the haplotypes using Supernova later, right. So basically each chip can process up to 8 samples, which is supported by a microfluidics platform, which is chromium, and it can do it very fast in less than 20 minutes. And then basically after you have prepared the library using the 10x genomic approach, you can proceed with the sequencing by any sequencer, short read sequencer such as Illumina or ion torrent, but Illumina combination is preferred because Illumina still dominates the short read sequencing market. Okay, and then after that the reads are assembled into contigs by various assemblers such as Supernova. Okay, so basically the DNA is bound to beads, and basically each fragment goes to one bead, and then the beads are differently labeled by different barcodes. Okay, so basically each bead or the DNA fragment will get a unique barcode, and later on sequenced by the short read Illumina sequencer, right. So you can see here, like this was say a DNA fragment, so it can be broken randomly. So for example 3 fragments emerge from is fragment 1 that contains a part of gene 1, fragment 2 that contains both part of gene 1 and gene 2, and gene 1, gene 2, or any loci on the DNA is like just for illustration purpose this is, and then a third part is coming from say another region which is kind of overlapping. So basically we can see that 3 DNA are overlapping, but many times this could be the real case. Sometimes you can miss the intermediate fragments also, like this one, fragment 2, and there will be gaps, and later on scaffolding another method have to be employed. So now each fragment, if you see by different colors, red, blue, and blue, right, are going to separate beads, right, and then basically each bead is basically uniquely barcoded. basically this fragment. So you can see that all the fragments that are generated from each of these fragments, right, are differently labeled by barcodes, right, and that is, and then when they are sequenced, each sub fragment will contain the same barcode which can be separated later, right. And also it will tell that because they are coming from a single fragment, the same type of barcode will have overlapping sequences or basically reads from that particular fragment of the DNA, right. And then basically based on this information of the barcodes, they can be assembled together into larger contigs, which basically was our starting point of the DNA also, right. So this is the theoretical situation, but many times there could be like gaps in between or places where we do not find any overlap, or because it is a random fragmentation, and generally we are doing up to like 56 or 60x coverage. So it is not possible that all the fragments will come, but still it is a good method to do long range scaffolding and also to resolve the haplotypes. Okay, so they provide a series of Python scripts, and basically the two are more important: the process 10x reads.py and filter 10x reads.py, which process the data that we generate from the 10x genomics DNA platform. Okay, and these reads can basically are to clean the data, right, because it contains barcode information, it contains the information from like primer which may be there, right, because each fragment was also amplified. Okay, so those have to be removed. Okay, and then basically it generates the FASTQ file from each of the the initial BCL to FASTQ, or it could be long range MKDA FASTQ, Supernova MK FASTQ from these files. Okay, so FASTQ files are generated by this script, and later on they are removed by another script to basically get rid of the barcodes and primers. Okay, so basically the input for the first program, process 10x reads.py. So so basically this will be a folder where you have installed the program. We have already seen in the previous lectures how to download and install this program, and then this is the program name. It's a Python script. Okay, and basically it has various options like -1 for the forward file, -2 for the reverse file, and -0 for basically the output folder that where you want the output to be. Okay, so basically this can generate the FASTQ files from like the initial BCL to FASTQ format and other FASTQ formats also. It will extract the GEM barcode, because basically Chromium is using a GEM technology, GEM. So it can extract the barcode that is there. So basically that is there in the first 16 base pair of the reads, right. Okay, and basically that is how it is improving both the sequence and this quality. And then it trims the read from one end. Okay, next 7 base pair of the read. Okay, and basically the extracted barcodes are compared with the list of barcodes which are already there, because it's a proprietary technology, so they are already using the barcodes for which they have the necessary information, and basically they list that which barcodes have been used and during the library preparation, and then the barcode which is there in a sequence is compared with the initial list of barcodes to see that which fragment is having what kind of barcode, and then they are compared in this way, like exact match between the barcodes in your sequence and the database or the white list of barcodes. A mismatch, basically partial match. Ambiguous means that they are not able to match, or unknown means that they could not find a relevant barcode, right. And then basically these reads are then further filtered and accepted based on match, mismatch and ambiguous barcodes. Using this program that uses that, basically using the previous information, it cleans up the file. So it is filter 10x reads.py. So for this it takes the input file, the list of the processed barcodes that is extracted from step 1. They R1 file, the R2 file read forward and reverse reads with the labeled like previous step generated processed output from the previous step, and the output folder name filtered underscore out. Okay, so basically this script analyzes the reads which are already processed by the process 10x py program. Okay, for specific barcode stated, that is what was the match, mismatch, ambiguous or unknown, right? And based on that it can filter, basically you can retain only the good quality ones, or you can also include the only the matched ones, or mismatch also you can give some preference. Okay, and you can take, but unknown you may like to reject because you do not know from where they are coming, right? Now this was one of the step to improve the or basically that is necessary to get rid of the barcodes and the primers, and okay, and to get a clean data. Then basically the reads you have filtered in this step, that was the 10x Genomics Illumina short reads, okay, with both forward and reverse and okay information, right? So now the idea is to assemble these reads, and the assembler that is used is Supernova, which was also provided by the same company. Okay, so it's a package for de novo assembly from Chromium link reads, right? So either you can use the previous script also, but if you do not use also, basically Supernova can also clean the data, and also can get rid of the barcodes, okay, in this step. And also based on barcodes you can segregate and also the assemble the contigs, assemble the reads into the contigs. Okay, so basically the key feature is that it creates deployed assemblies, okay, which was absent in other software, because in other programs basically the places where there was heterozygous sequences, they had no way to resolve those, but here you can generate assemblies which could be deployed, okay, and therefore it can provide information about both the maternal and paternal chromosomes over very long distances. So Supernova and basically the 10x reads cannot help you to generate like the complete chromosomes or continuous assemblies, but they can help you to at least do better sequencing for DNA fragment wise, and later on using the more Illumina data or the nanopore data, you can still make it a more contiguous assembly. We will see some examples also in the end. So basically the other methods merge homologous chromosome to single incorrect consensus sequence, but here it can try to resolve the haplotypes and also create a deployed assembly. Okay, so basically as I mentioned that the barcoded 10x data provides a shallow coverage. It is just something like up to 50 or 60x coverage, because actually this Supernova software also cannot handle more data than that. Okay, we will see that how many million reads it can handle, right? So basically that is why it is not meant for generating the complete chromosome, or but it makes large contigs because it is containing long distance link reads and also like long distance scaffolds also can make, right? And basically it is also good that you can see that, for example, where many contigs are at least few kb long, because the fragment sizes were very large, and most molecules pass through a given contig will have at least one read landing on it. So that means it is getting the evidence from the reads that are mapping to those contigs. Okay, so if you see this, we will see some pictures also to understand the Supernova methodology better. So the minimum requirements to run Supernova is that you should have a 16 core or more Intel or AMD processor. So basically in 16 cores there can be like up to 32 threads that you can use, or preferably like 25 to 28. Even more threads are better, because it is supporting the assembly, multi-thread assembly. Okay, it requires large free disk space, right, because it depends also upon the genome that you are going to sequence. Okay, and also a large amount of RAM, 256 GB or above, because this process is memory intensive and it requires and sometimes it also recommended to have up to like 1 GB of RAM, 1 TB of RAM, which is recommended, right? So this is the paper that discusses the importance of supernova and how basically it can do the deployed genome assembly or construct a better assembly. Okay. So if we see this, basically the problem with the previous software was that it was not able to resolve the places where the alleles were maternal or paternal or heterozygous. Okay, but this software can resolve this. Okay, and in the previous attempts only a few times it was possible to have the true deployed de novo assemblies. Okay, but that was very expensive and time consuming, but this software can achieve that from the link reads, that data that we gave. And the two that we are talking about was, one was using Sanger and one was using thousands of clone pools, which is a very time taking intensive process, right? So basically they demonstrated Supernova as a low cost method for creating true deployed de novo assemblies. Okay, and it can use only 1 nanogram of high molecular DNA, which is very less DNA requirement compared to the other library preparation methods, and that is also the advantage of this method, right? And uses a 10x genomics microfluidic platform to partition the genome. So basically they used this technology for 7 human samples and generated low cost high-seq data. So basically they prepared the library and then did the high-seq genome sequencing. Okay, and then carried out the assembly using just one script, you can say push button algorithm, Supernova basically. And each computation took about 2 days on a single server and yielded contigs which are more than 100 KB long, which is a good basically you can say contiguity of contigs. Okay, and scaffolds were even longer than 15 million base pairs, which is also a good number, right? And the basic capability was to determine the actual deployed sequence in a sample, which was not possible by the other methods. Okay, so like in this picture you can see that this is the de novo assembly by Supernova. So this line indicates places in a assembly where the sequences are homologous in the two chromosomes, right? Where okay, but at these places this this indicates that there are two possibilities, or there is one parental allele at both locations, right? And which forms a bubble, right? So basically you can see that it is also coded by two yellow, golden or blue bubble, right? Which so basically this is an edge and this is a bubble. So this is okay, the and attempt to resolve the places or identify the places where the sequence is differing in the two maternal and paternal chromosomes, right? And these edges are the places where they are not differing, or basically those are the homozygous, right? And then it tries to recover the deployed genome from the assembly. So Supernova can be downloaded from GitHub or from the 10x genomics site. GitHub you have to check if it is available, still available there, or right? So but Supernova can be downloaded from support.10xgenomics.com, de novo assembly software downloads, right? By filling up or subscribing to the a simple agreement, right? So it generates phased whole genome de novo assemblies okay, from the Chromium prepared library. So one of the requirement is that it should have at least 38 to 56x coverage of the genome, right? Below that, if it is less than 38, it will not work. If it is more than 56 or 60x, basically the program again will exit because it is finding too many reads, right? So both the coverage as well as the number of reads matter. So that is why if you have a very large genome like more than 3 GB like human, okay, then more than 4 GB I think, so then the number of reads to achieve that coverage for that 4 GB genome will also be higher, but it has a limitation of the number of reads also that it can consider, because it's a memory intensive program also, so it will not work or basically as expected for longer, even if it does not exit from that. Okay, so higher coverage is better, but still there is a limit. It will exit if it finds the coverage is far from the recommended range, that means it is, you have provided too much data to it. Okay, and at most 2.14 billion reads are allowed. So your genome size and the coverage, okay, and the number of reads together will get you the number that how much you can do with this, right? So basically more than 4 GB or more than 3 GB you can say, okay, should be considered experimental, that means it may not be supported by Supernova. Okay, so it has two processing pipelines and for processing. Okay, first of all, Supernova MK FastQ, which converts the BCL to FastQ of Illumina into basically demultiplex chromium prepared files. Basically the FastQ files from the BCL to FastQ, which is the system format, right? So it recovered the FastQ format files from those based on also on the number of barcodes and type of barcodes and resolving the barcodes and the primer that were there. So basically then it takes Supernova run, which takes the FastQ file containing barcode reads from Supernova MK FastQ and generates a graph based assembly that we have seen earlier, that is the edges and the bubbles, right? And then using the graph based assemblies, it creates several kinds of FASTA files for downstream processing like pseudo-hap2, like that we will discuss in the next steps. Okay, so basically it required the sequence ID, that means a unique ID that you want to define to your sequence, the path to the FastQ files. Okay, basically if you want to select in that path only one sample or multiple samples, you can specify, okay, how many reads you want to target, okay, or consider for the assembly. So even if you have more amount of data, you can specify that, okay, use only these many number of reads because there is a read limitation, right? Or if you don't say, it will take all maybe, right? And the local how many cores you want to use to run the Supernova. So depending upon the cores in your system, you can select this option, that how many cores you want to dedicate. More the number of cores you have in your system, faster it will do. Okay, and the memory, again it is depends upon your system's memory, but at least 256 GB should be given for larger genomes, and more is better. Okay, now how to estimate the number of reads that you should have. Okay, considering the length of read is 150 base pair, and basically your genome is about 800 MB or like 0.8 GB, right? So basically the 56x coverage that you want to achieve with this genome when you are preparing for sequencing or ordering sequencing from a third party. So basically you have to divide the size of genome by the length of the read multiplied by 56, that means 56 times of that. So you will reach to a number of 298 million reads. Okay, and you can select the max reads is equal to this much, basically, which is the 56x. So even if you have more reads that you have generated, you can still restrict it to this number. If you have less than this many number of reads, basically Supernova will if basically your coverage is less than 56x or the number of reads it can handle, so you can give all also. So it will you don't have to bother about restricting the number of max reads here, right? So basically in that case Supernova will use whatever is available, and maximum it can go for this many number of reads, which is after, if it is more than that, it may exit, right, and it will not do the processing. Okay, so basically Supernova output is dependent upon the it is trying to resolve the deployed genomes. Okay, so you can have different styles of output. One is like the raw style, where every edge in the assembly is given as a FASTA record, like this. These are the edges. So basically you will have like too many sequences and which are short also, right? And so generally like the pseudo hap style is more preferred, right, which is this one. So here you have two options of pseudo hap 1 and pseudo hap 2. So basically pseudo hap style, for example in this case it will connect the edges and the bubbles in this way, right, and it will generate only one output. Like this way it will read, okay, and whatever is coming, the sequence coming over here, it will just connect the two all the sequence and generate a single output, okay. But if you give pseudo hap 2, it will generate both the basically deployed assembly, and basically it will generate two parallel pseudo haplotypes in two different separate FASTA files, okay. In the first one, pseudo hap, it may mix the maternal and paternal allele, like you can see, like yellow is here, then again yellow is here, and yellow is here, okay. So it is connecting the edge, which is the homozygous regions, then the diploid region, or basically the region which is heterozygous between the two chromosomes, okay. So yellow, then blue, and then yellow, that means it is mixing the maternal alleles, maternal paternal alleles, whereas here it is trying to generate the two paternal alleles separately. Okay, so to run Supernova, basically you can say Supernova run, give the ID of the assembly output, is assembly output folder name. Just give some to your samples like NT or BT, or basically just one sample if you have just one, okay. Input sequences, that is input 1 and input 2, that read 1 and R1 and R2, right. So okay, so fast QS. Maximum number of reads you can specify all, or you can limit the number of reads that we discussed earlier, because it can handle only 56x or maximum of x number of reads, so you have to restrict to that number, right. So if that your reads are less than what it can process, then you can say all, but if it is more than that, then you have to specify that how many reads from the total number of reads it should use. So restricting it up to 50x also, 56x also. How many cores you want to use, like 40 in this case. Memory 950 GB, it is specified, for example here, okay. And then basically in the from the output, basically you can select that you want pseudo hap 2 output or pseudo hap output, okay, and then give the assembly directory which was generated from the previous step, like from here, okay, and outs and assembly, okay. And then the another output file which is pseudo hap 2 output, where it will be stored, okay. What should be the size of minimum contig that you want to use? So if you say 0, that means you want all the contigs that are generated and you are not putting any threshold for the minimum contig length to be stored, right. So that is Supernova, which is a very good tool for creating assemblies using the 10x genomics data, okay. Now genome scaffolding, like we have discussed so many tools so far, that is using the nanopore data, link reads data, or the Illumina reads data. So now now basically you have created their separate assemblies, okay, in the contigs, and those programs have tried to depending upon their source data, that is nanopore, link reads, or Illumina data, they have tried to generate the contigs or achieving some N50 or BOOSCO coverage, okay, a number of genes and like that, if you do use only one method. Even if you are doing hybrid, so basically you have to produce scaffolds from those contigs, okay, and for which you can also use the RNA-seq data by Agouti, that we'll discuss later. So we will show, or basically we'll discuss a few algorithms that can help you to scaffold your contigs further, okay, preparing a better or much higher contiguous assemblies, and also how the RNA data can help you to improve the assembly, okay. So basically scaffolding is a process of linking together non-contiguous series of genomic sequences, that is contigs, right, into even longer sequences where some regions could be unknown and can be like for which like ends will be placed, but still at least you know that these contigs belong to this sequence their order basically is also, it is, because while doing the assembly, the order or their orientation can be here and there. So basically you are putting them in the right order, into the right orientation, into a scaffold, okay, and connecting long distance contigs also based on the information that you have from reads or RNA-seq data, etc., and then filling the intermediate regions as ends, right. So basically it will read the data from the previous assembly or contigs, the raw reads also, okay, and additional information like RNA-seq that we will see in the case of Agouti, okay. So scaffolding is basically can be done for long and short read data, and using this scaffold, the transcriptome data, to achieve the higher or better contiguous assembly, okay. So generally, like short read or next gen sequencing, which is long read, can be used, but those are generally fragmented contigs are generated out of only if you use long read or short read data. Generally for the short read data, the contig size will be much smaller. So scaffolding is a step that is very much needed to first, if there is a misassembly or putting the contigs in the right orientation and order, right. So basically scaffolding will achieve this job. Okay, so one of the software for like 10x genomics scaffolding is data scaffolding is ARCS, okay, which uses the link read data and the long read sequencing data together to do the scaffolding, okay. So basically in this, if you run to run this program, it is like ARCS make and then ARCS draft genome assembly, okay. So basically this is the genome, okay. So basically you will only say draft equal to the genome assembly, and here you are mentioning the file that contains the previous genome assembly that you have done using the short reads separately or the long reads and like that. So like this will be genome assembly.fa, okay. Reads equal to barcoded. So basically the barcode that you have used or in the previous step or the 10x, okay. So that is this barcode.fq.gz. You don't have to specify .fq.gz, it automatically consider those files, okay. And the minimum contig length, which is 0 in this case, okay. So basically it tries to pair the sequence within a draft assembly and then creates the layout of those for the scaffolding purpose. So for this it is creating alignments in BAM formats and using the set of pair reads from the same barcodes. So basically the cases where reads which are coming from the same barcode of the same fragment, if they are aligning to different contigs, that means from a single region that was supposed to be coming from the same DNA fragment because it had the same barcode, okay, you have created two contigs, whereas those two contigs actually belong to the same DNA fragment because we know from the barcodes that we have checked, okay. So that means those two contigs which contains the same barcode reads, okay, are disjointed because there was some missing region in between, okay, right. So for example, this was the region and say this barcode was there, right. So there could be one region like this one, okay. So basically your reads can map here and here, but still they are coming from two distant contigs and they should be connected together, and also their orientation and the order, right. So that is decided, and then they form the link between the two contigs, okay, by using certain amount of evidence of the read pair that are aligning to those, okay, and then linking those together as scaffolds, okay. So basically ARCS software can be downloaded from the GitHub site, and it has provided a lot of improvement. For example, from a ABIS, which is a de novo assembler for the short reads for the Homo sapiens human genome assembly, was improved almost 6 folds, okay, using a moderate amount of chromium data. That means just using 25x to chromium data of human genomic reads, it could increase the assembly contiguity by almost 6 times or 6 folds, which is a good achievement, okay. So basically it is deriving or creating k-mers from the contigs that you have already generated from the previous assembly, okay, and then from the, it is also taking the raw reads, okay, and then mapping the barcoded or the reads which are coming from the same barcode to the k-mers that you have generated from the contigs, and based on the evidence of where that pair of reads are mapping to the different contigs, they are assembling the contigs together into longer scaffolds. Okay. So, we will not go into details of this algorithm, but basically I have mentioned the concept that it is using, because that details may be out of scope for this purpose, okay, for this series of lectures. Okay, and we have also discussed how to use that program. The another scaffold that we want to discuss is the links or long read scaffolder, okay, which can use the nanopore sequence data and the other error containing sequence data such as PacBio to scaffold or generate high quality genome assemblies. Okay, so nanopore or PacBio data can be used here. Okay, so basically it works like this: links and the genome.fasta, or the file that contains the genome. Okay, the file with read location, that means where the reads are you are storing. Okay, that is the output prefix that you want to put in front of the files or the output folder. Okay, number of threads you want to specify, minimum contig length. Okay, so basically more details are here, like F is the assembled sequences to scaffold, that is the multi-FASTA format with single with each sequence a single line, okay, that you have constructed from the previous step. Basically in -s you will provide the path to the long sequences reads file, the individual file, the previous one the assembly, the single read, basically the long reads. So it is using both the assembled data and the long read data together, because it will do the mapping and alignment using the k-mer approach, right. What name you want to specify to the output folder, basically some kind of tag you want to specify, number of thread, minimum contig length to consider for scaffolding 0 or above. So basically giving 0 means consider all, right. This sliding window, step of sliding window, because you are generating the k-mers from the long read, so you may specify that you can like generate k-mers with one step, okay, like this, okay, shifting one base pair or two base pair or three base pair, like that. And -l minimum number of links or k-mer pairs to compute scaffold, that means what evidence or basically how many number of links or k-mers pair that should match you should consider while you are doing this scaffolding. Okay, so this is more details can be found in this paper, which is published in Giga Science, the links, scalable, alignment free scaffolding of draft genomes with long reads, right. So basically if you see here, it was for an ABS assembled K E. coli genome, it could increase the assembly by almost 5-fold by use of the long read data. Okay, and so basically whenever you see a paper, they always show that how that algorithm or the method improved the previously existing assembly. So in this case they have demonstrated on a E. coli genome which was assembled by ABS, and now they have improved the assembly by almost 5-folds by using the links. That means they are using the previous assembly, the nanopore data, and then comparing the two using the k-mer approach or mapping the read pairs to this, okay, which is mentioned here. So for example, it has two main stages, a contig pairing and a scaffold layout phase. So basically in the contig pairing phase, they are matching the reads to various contigs. The k-mer pair, okay, are uniquely placed on contigs are identified, so that if the k-mer pairs are placed on different contigs, that means those two contigs belong or coming from the same DNA fragment and should be connected together. Okay, if the given distance between the two contigs is also appropriate and justified by the k-mer pair distance, right. Okay, and then they also put the gaps or ends in between at the scaffolding stage, and then use those scaffolds are built using contigs as the seeds, right. So links can be downloaded from GitHub, and generally GitHub is the place where you download most of the software. In case of 10x genomics software, Supernova and 10x algorithms, those should be downloaded from the 10x genomics site because those are proprietary. Then after scaffolding, you can also use after the read-based scaffolding, genomic read-based scaffolding, you can also use the transcriptome read-based scaffolding of the previous assembly, right, which can be done by Agouti, which is annotated genome optimization using transcription or transcriptome information. That means you are using the transcriptome data which is coming from the transcribed exons, or basically the genic regions which are protein coding genic regions. Okay, so you are using that along with the gene prediction file and the previous assembly to improve the previous assembly, improve the gene structure by adding more exons if they are missing, by connecting the genes, or improving their start end, right. So basically it uses the RNA sequencing data, okay, to simultaneously combine contigs into scaffolds and fragmented genes into single genes by using the RNA-seq information. So it increases both the contiguity of the genome and accuracy of the gene annotation also. So basically the input will be the genome in FASTA format, the paired and RNA-seq file, okay, which are basically mapped to the previous assembly using in BAM format. And you also have to do the predictions by Augustus in the initial assembly in GFF format, because that will tell that what are the coordinates of exon intron which the Augustus has predicted, right, in your assembly. So it can accept both these contigs and scaffolds. In the scaffolds, basically if they are too long, it will break those into at certain position locations into again contigs, because in scaffolds you have tried to put the contigs again into a longer distance assembly, or basically connecting the contigs together to scaffolding. So it derive the contigs again from those scaffold and create a split assembly, and then basically again do the scaffolding using the RNA sequence information. Okay, so basically Agouti will give you an updated genome assembly and updated set of gene predictions in GFF format, because now you have used the transcriptome data also to improve the gene structure and also to check whether the previously existing scaffolding or the contig arrangement or orientation was correct or not. So that's why Agouti is a very good addition, or basically step that you should do when you have the transcriptome data. You can use your own transcriptome data, or you can also download the transcriptome data from literature and other for your genome from other previous published papers also. You can also use the transcriptome data from multiple tissues, because it may not be possible that all the genes will express in all the tissues. So that is some ways to improve your genome assembly. Okay, so basically it will identify the joining pairs based on the RNA-seq reads that are mapping to different contigs. Okay, so there will be 4 situations which we will discuss in the next step. So basically Agouti can be downloaded from here, and this is the paper that is published to support Agouti. Okay, so they could improve the previously existing assembly and using the transcriptome data, and provided a proof of concept for that. Okay, so like in this picture we can see that how it actually doing this job. So basically there are 4 cases discussed here, A, B, C, D. So in case 1, basically there are 2 contigs which are joined by Agouti because they carry exons of the same gene. So basically since basically if there are 2 contigs that carry the exons of the same gene, right, they should be connected because they are part of the same gene. So basically 95% of the contig pairs scaffolded by Agouti fell into this category. That means they can be connected, they are basically coming from the same gene, right. Case 2, where only one end of the joining pair overlaps a predicted gene on either contig. So that means only one end is joining, like you can see in this case basically both are the read pairs in the case 1 were mapping to the same gene. Okay, in case of 2, basically it is mapping to some region in between the read pairs of coming from the RNA sequencing data, which means that in between your predicted gene there is one more region which can be a new exon also, because you have the transcriptomic evidence or data for to support that. Okay, that means there exists a new exon in one of the predicted genes, right? Case 3 is that the joining pairs are mapped on two different annotated genes in the same genome. That means RNA-seq data is mapping to two different genes. Okay, that means those two genes are actually not two genes, but they are called as two separate genes, and they should be merged into a single gene. So basically, this other that two genes should be merged into one, and they were the failure of, or basically there could be cases of transcription termination failure also, and okay, which are complicated cases beyond discussion here, right? But basically the point here is that the genes that were basically annotated separately or basically called twice should be merged into one if you have evidence that your RNA-seq data or the read pairs from the RNA-seq, okay, pair forward and reverse reads are matching to different separate genes, right? And the case 4 is that the joining pairs are not mapped to any predicted genes, which indicates that there is some region which is not called a gene, and there could be the existence of another gene, right, which is not predicted by the Augustus. Okay, so that means like for in this region it was this RNA-seq paired and data, the joining pairs, and those are not mapping to the previous gene, and okay, so that means there could be some gene which may be existing in between. Okay, so basically this way it is improving, first of all assuring that the gene structure is correct, finding information about new exons, merging the two genes which are called twice and basically are the same genes, and also finding new genes or evidence for new genes between genes, right? So that is how like Agouti they have done like 6 simulations using the contig pairs that they have generated from the scaffolds, right? And in all the cases they could see that in increase the scaffold N50 number of genes, improve the number of genes, right? Decrease the number of scaffolds in the assembly, because that means that you are connecting more and decreasing the fragments into more contiguous assemblies. Okay, so basically the thing that we discussed in the picture is also mentioned here, that most of the genes or 95% of the genes joined by Agouti, okay, will have the joining pairs mapping to the same gene, right? In some cases you will have the mapping pairs into a separate gene or in between genes, so those could be cases of new exons or case of merging the two separate genes into a single, because basically the RNA-seq data is suggesting that, or there could be a new possible gene which is for which you are have the mapping joining pairs mapping in between two genes, right? Okay, so there are other softwares also for scaffolding, and basically people like to improve the previously existing algorithms or finding new concepts for scaffolding based on the new data or algorithm or the logic. Okay, so if you search you can find even more, so but some I have mentioned here like SLR's super scaffolder or RSCAF or Plantus allele. These are the other scaffolders which you can try. Okay, so now you can see that basically usage of genomic and how the scaffolders or the RNA-seq data has led to the increase in the assembly contiguity. So basically you are dealing with short reads, okay, which do not provide a good contiguity or good N50 or Busco values, but they have lesser errors and important for the assembly, okay, for which you can do a De Bruijn-Gras based assembly and using so many assemblers which are available on this approach, okay, or you are using the long reads which are providing you longer you can say sequence regions of the genome, right, which can be connected by OLC method or overlap layout consensus method, and so many softwares are available to assemble this also. Or you can have a hybrid assembly approach where again you can do the long and short read assembly separately or together, and also using the RNA-seq data that we have seen by Agouti or other methods, and creating better or high quality scaffolds, right? So basically RNA-seq data can help you to improve the assembly, find new exons, missing exons, alternative splice isoforms, or basically missing genes also, right? So together, like if you see, like once you have an assembled genome, you would like to correct it for errors by using both long and short read data. You would like to correct it for the scaffolding and orientation of contigs in the right order and orientation, improvement of the gene structure using the RNA-seq data, closing the gaps as much as possible using the read data that still can be matched to the assembly that you already created. Then again, going for a polishing step, you would like to correct the errors using the preferably the short read data, which is more accurate, okay, and having the final assembly for further analysis, right? So for one genome, for example Jamun, you can see that using a different method that we have discussed today also, like after the polishing step, if I use Agouti, which is using the RNA-seq data, it decrease the number of contigs, right, and also decrease the number of scaffolds, right, improve the number of genes, and also the Busco completeness was also increased slightly, okay, which means that Agouti, or basically this using the RNA-seq data is helping. Then using ARC scaffolding, you could further decrease the number of contigs to a large extent from 9500 to 7800, which means that more disjointed contigs could be connected together in their right orientation and order, right, into more contiguous assembly, right? And like we have shown for like single contig, all contigs, or up to more than 10 kb contigs and like that, right? There was no change in the GC content because basically GC content is for the genome, and this assembly using the RNA-seq data or scaffolding may not change that, should not change that, right? Then ARCs also increase the Busco completeness by some percent, by some fraction, which is, and anything after like 98, 98.2 or 90.3 is like still improving, right? Then linked scaffolding also decreased the number of contigs. So basically you can use either this or this, right? And also decreased, improved your Busco score, right? Then LR graph closure is another software which also can be used, right? And basically finally what you achieve is much different from the initial assemblies, right? And or basically more acceptable and more contiguous assemblies by using multiple types of scaffolders and assemblers, which could be read based or RNA based, RNA sequence based. For the turmeric genome, if I had done only the 10x assembly using only the 10x genomics data, the achieved N50 was 15.8 kb, okay, because as I mentioned, it is not very useful for a highly contiguous assembly, right, or making very long contigs, okay, but and then for that basically nanopore or PacBio data are used. So just from nanopore data you could get 60 kb N50, that means more contiguous assembly or longer fragments in your assembly, okay, and by using both of them together you could achieve N50 of 100 kb, which is a very good improvement over using either of the short read or long read data. So basically hybrid assembly performed better. So basically from the Supernova assembled raw genome, the complete Busco was 75% from, that is using the 10x reads. For the nanopore read using the fly, the Busco was 71%, whereas using both together it reads 92.4%, which is a huge improvement on like either doing the two separately. So scaffolding definitely helps, right? Similarly, fragmented Busco scores also decreased. That means it was more contiguous then. Okay, missing Busco also decreased. That means you are not missing much of the single copy ortholog genes, SCOs, right? And basically totally like you were searching for 1614, that is using the reference that you used during the Busco, basically running while you running the Busco. Okay, basically, so you can see that scaffolding and basically improving assemblies using both, connecting both the long read or link read data or only the single read data always improves the contiguity and the Busco values of the assembly. So in this lecture we looked at the 10x genomics data, the assembler that can be used to assemble the 10x genomics data Supernova, and how to clean the reads, and then the different scaffolder that can be used to improve the contiguity, decreasing the and reducing the disjoint assembly into more contiguous. RNA-seq data can be used to improve the contiguity as well as to improve the gene predictions, and also connecting the further improving the assembly, right? And some examples of how it changed the assembly in some of the previously published papers. Okay, so this was good for today. Thank you.